All right, a quick run through on photosynthesis so that we're ready to talk about this in class this week. So chapter 10 is about photosynthesis. This is, of course, the other half of the circle of life. And I think for most people, cellular respiration tends to make more sense because it has to do with us. But of course, we can't do cellular respiration without food, and that is going to be provided via photosynthesis. So, this is, to me, maybe one of the craziest things in all of biology, right? We are looking at, in this process of photosynthesis, how sunlight builds, right? The trunks, the leaves, the branches here of this tree, but also, the seeds and the nuts and the leaves and things that we eat, right? And so what we're going to see as we dive in here, right, is that plants and other photosynthetic organisms contain chloroplasts, right? This is a specialized organelle where photosynthesis is going to occur. Photosynthesis is the process where we convert solar energy into chemical energy. Blows my mind, right? We're taking energy from the sun and we're putting it into energy in these chloroplasts, which then hand it to the plant. And really, directly or indirectly, photosynthesis feeds almost the entire living world. So when we talk about producers, organisms that do photosynthesis, we often refer to them as autotrophs, like auto-self, like self-feeders. These are going to be organisms that sustain themselves without eating anything. Again, wild, right? But that's what plants, algae, cyanobacteria, etc., are doing. Right, so they don't have to eat anything from other organisms. These are the producers of the biosphere. They take carbon dioxide and they turn it into organic molecules that other organisms can use. Again, super wild. Here's some other examples, right? Most of these autotrophs are going to be photoautotrophs, so actually getting the energy for this. What word do I want to use? I'm looking for an adjective for how phenomenal photosynthesis is. Most of them are going to be using energy from the sun. There are a few examples of like deep sea vents and things where organisms can get their energy from other chemicals. We're not going there in this class, right? We're going to stay on land, okay? And again, if you're like, I don't really care about photosynthesis, remember, other organisms, not just these beetles, but us, benefit from photosynthesis, right? Because we are, if plants are autotrophs, we and these beetles are heterotrophs, right? And heterotrophs have to get organic materials from other, right? Other. So that we have to obtain organic materials from other organisms. Heterotrophs are called consumers when we look at them in the biosphere. Now, some eat other living organisms, right? These guys are eating this plant that's still alive. Others, things like decomposers, are consuming dead organic material, wastes, feces, things like that. Basically, all heterotrophs, including humans, depend on photoautotrophs for food, right? So we are absolutely dependent on photosynthesis, even though we probably don't spend a whole lot of time in our daily lives thinking about it. Here's your chance, right? We're in college, we're thinking about things we haven't spent time thinking about before. Okay, before we dive in there, one thing to think about, so most of the fuel supply that we're using right now on Earth are from fossil fuels, coal and oil. This coal and oil, if you haven't heard this already, are really just the remains of organisms that died hundreds of millions of years ago, right? And under pressure, those dead bodies, under really exacting environmental conditions, became these deposits of coal and oil. Fossil fuels are being consumed faster than they're being replenished, right? They are a finite resource. And so a lot of people are starting to think about alternative fuels, right? What can we do to replace coal and oil? And this is one of the ones that's actually really promising and I think interesting. The idea of being able to use algae to produce what we would then call biofuels. And you can make biodiesel, you can the really fun thing I've seen with this is trying to build them at coal burning power plants, right? Because the waste product of that power plant, CO2, which we don't necessarily want in the atmosphere, can actually be kind of bubbled through these tanks, and it's food for the algae, right? And they can actually then convert that back into lipids that we could use as fuel. Anyway, kind of fun. Okay, so let's jump in then and talk a little bit more about photosynthesis. So just to give you a framework, right, of where this is going on, we're looking at this, of course, happening primarily in leaves. We're going to focus in on plants more than algae and things. So we think about this happening in leaves. And then as we zoom in kind of on a cross section of the leaf here, we'll see that the leaf has an organelle called the chloroplast. Chloroplasts are actually structurally similar to photosynthetic bacteria. And so they fit under this theory that's referred to as the theory of endosymbiosis, that these were actually once free-living photosynthetic bacteria. They were engulfed maybe by an early eukaryotic cell, and they developed this mutualistic relationship, right? The chloroplast, that bacteria, was producing sugars, right? And then the cell gave it some kind of protection, and so they hung out. Anyway, there's a lot of evidence, right? The structures are similar, the DNA is similar. Interesting. Okay, if you look inside of that chloroplast then, and I will say these chloroplasts are mainly found in cells called mesophyll cells. Sorry, this is the cell. And then inside of the cell, we see chloroplasts. That's what all these like little individual spheres are. And you actually saw these chloroplasts. I think one of the early labs we looked at a plant called Elodea it was in the microscope lab. And so inside of that cell, we can see all of these chloroplasts. These cells, again, typically are inside a region called the mesophyll. So this is like the interior tissue of the leaf. Anyway, gases are going to enter and leave the leaf through a pore. This pore is called a stomata, like a little mouth. And that's going to be important, right? Because we're going to need CO2 to come in to provide the fuel for photosynthesis, the carbon, right? The matter for photosynthesis, I guess. And then oxygen is the waste product, right? And so we actually need that to leave. So when you look inside the chloroplast, it actually has like a double membrane, just like we saw in mitochondria, and it's filled with kind of a thick fluid called the stroma. So stroma is a lot like cytoplasm, but maybe thicker. What gets frustrating here is stroma and stomata. Don't confuse those, right? Stomata are the pores. Within the chloroplast then, sitting in this stroma, are these stacked structures called thylakoids. And they're these little sacs. Each of the little individual circles that you see, sorry, there's a zoomed in picture in your textbook, but each of those individual circles is a thylakoid, and the whole stack is called a granum. Okay. Inside of each of these thylakoids is a pigment called chlorophyll. Chlorophyll is the green pigment, and this is going to be super important in photosynthesis. So just a framework, right? Obviously in plants, in chloroplasts, inside thylakoids. Thylakoids might be the new one for you. Okay, so this, I do think it's kind of interesting. As we look at photosynthesis, we kind of looked through this same equation with cellular respiration, talking about oxidation and reduction. And so I think this is kind of fun. It's the exact opposite. And this time we see CO2 that a plant is taking in from the atmosphere getting reduced, right? It's taking those hydrogens and becoming a sugar molecule. The water that the plant uses is where those electrons, right, where the hydrogens are coming from that get stuck onto glucose. And so water is actually being oxidized. It's getting the electrons ripped off, and here we see the formation of oxygen. Now, researchers actually broke this down and followed where these different molecules were coming from. And this is actually part of how we understand photosynthesis to be taking place, right? Is that we actually see, if you say use radioactive dye, you can follow that the hydrogens from the water are what's actually reducing the CO2, right, to glucose. And then when we look at the oxygen byproduct, that's coming from the water. And then the water byproduct that comes out of photosynthesis is also a mix, right? So it's actually getting the oxygen from CO2 and hydrogen from water. Anyway, I think that one's kind of interesting, right? But there's our equation that we're more familiar with. So make sure you know who's oxidized and who is reduced. All right, so when we look at photosynthesis, it's going to be in two main steps. The first one is called the light reactions. The second one is called the Calvin cycle. But we're going to step back here. So here we are, we're inside of our chloroplast, right? We're in the stroma. Here, each of these is our thylakoid. The whole stack, again, is the granum. So what I want you to know here is that in the light reactions, right, notice that light is providing the energy to essentially split water. Here we see the oxygen part of water coming out. The electrons, the hydrogens, are being added to a molecule called NADPH, right? This is an electron carrier, just like we saw in cellular respiration, but instead of NADH, we get NADPH. So I remember it's photosynthesis. There's a P in there. So NADPH is going to be our electron carrier, right? So we see NADP coming into the light reactions. It gets reduced. We also see that during the light reactions, some of that energy goes directly into phosphorylating ADP into ATP. So in the light reactions, we get ATP, NADPH, and oxygen. Okay? Again, to me, this is the craziest thing. Plants actually split water into oxygen and hydrogens, right? The oxygen comes out as a waste product, which we love, right? And then the hydrogens are going to get carried on to the Calvin cycle. Okay, so this isn't so crazy. Don't worry, a little bit we'll go into more detail. It gets a little crazier than that, right? This is big picture. So here comes the Calvin cycle. And we will go into more detail here, but basically the ATP and the NADPH are going to power the Calvin cycle, right? The ATP is the energy that we need in order to run this anabolic reaction. Anabolic reaction. It's also anaerobic, but that's not what I meant. Anabolic, right? We're taking a simple molecule like CO2 and we're going to make sugar, right? That is requiring energy. Sweet, we got energy, we got ATP. And also notice basically the difference here between CO2 and sugar are some hydrogens. We need to reduce CO2, and we are in fact going to use the hydrogens from NADPH. Right, so again, that's your big picture. If we go deeper and you get lost, come back to this. Make sure you know what's going on in the light reactions and the Calvin cycle, and we will break this down a little more. So this light energy that's feeding photosynthesis, it actually, this gets quite a bit actually into physics, right? So the energy, light, I guess is how I should start this. Light is electromagnetic radiation, right? So sometimes you'll see EMR, electromagnetic radiation. And electromagnetic energy is actually traveling in waves. And this is hard for people often to picture, right? Because again, if you talk to physicists, it's interesting because it's also kind of these discrete particles called photons. Anyway, what you want to know about light here are a few things, right? So it is traveling in this wave-like fashion. And if you measure from peak to peak, right, peak to peak, that's called wavelengths, right? The distance between two crests or the distance between two troughs, that's the wavelength. And the wavelength really determines what kind of electromagnetic energy you have. So here you see different distances, right? These are longer waves. Let's draw this out, right? So, right, they're longer waves, right? The distance between here and here. And over on this end, I can do this. Terrible drawing. right? It's a shorter wavelength. Now, the big thing here is shorter wavelengths actually provide higher amounts of energy. I'm like just totally in the way here. I'm going to go right in the middle. So shorter wavelengths provide more energy, and then longer wavelengths have less energy, right? Electromagnetic spectrum is this entire range of electromagnetic radiation, but what we're mostly interested in here is what we call visible light. And so this is going to be light that's between the wavelengths of 380 nanometers to 750 nanometers. That's what produces colors that we're actually able to see. Visible light also includes the wavelengths that are the ones driving photosynthesis. So again, if you think about, do you guys remember Roy G. Biv, right? Red, orange, yellow, whoop, there's yellow, green, oh, I'm not showing it on there. Red, orange, yellow, green, blue, indigo, violet. Do you guys remember Roy G. Biv? Anyway, violet, shorter wavelength, higher energy. Red, longer wavelength, lower energy. So when we look at photosynthesis, right, you may have pestered your parent at some point in time. We'll get out of the way again. Pestered your parent at some point in time and said, why are leaves green, right? And maybe they didn't have an answer. Now you will have an answer. So what ends up happening here is that the chloroplasts in leaves are filled with a pigment that we already mentioned called chlorophyll. And pigments actually absorb different wavelengths. Wavelengths that aren't absorbed are reflected, right, or transmitted. And so leaves actually appear green because chlorophyll reflects green light. So then you could start to say, oh, well, that means that chlorophyll can absorb maybe the other ends of the electromagnetic spectrum, right? Here and up here, but this green is just getting reflected, right? And then when it hits our eye, the plant appears green, right? Okay. And so you can, of course, test this. We have equipment in certain labs where you can actually shine light that's kind of being separated, right? It's going through a prism, so we can see all those different wavelengths, and then we can specify that only, right, green light is passing through here. If it passes through, right, if it's not absorbed, we can tell. So they're trying to lay out here what we call an absorption spectrum. If that particular wavelength of light goes through, it's not being absorbed by the plant. And so here we see, right, chlorophyll does not absorb green. But now we're changing our setup here, so we're only letting blue light through. Notice that not near as much of that light is hitting this receptor. And so we would say, oh, chlorophyll actually absorbs that wavelength. This is what it ends up looking like. And so in this image here, what we see is that there's actually multiple different pigments inside of our chloroplasts. So notice there's chlorophyll A and chlorophyll B, and then a separate group of pigments called carotenoids that are generally considered accessory pigments. Chlorophyll is the big player here. So chlorophyll A, chlorophyll B are slightly different, right? The protein shape is slightly different that allows them to absorb slightly different wavelengths of light. But notice what happens here. Chlorophyll A and B and the carotenoids, right, are absorbing tons of light over here in violet blue. And they even absorb over here some in kind of that red light. But notice what they really don't absorb, right? Really in that green and in that yellow spectrum, right? And so when you put it all together, all the pigments, sure enough, we see them utilizing, absorbing lots of that energy in the blue violet and in the red. Interestingly, carotenoids during the fall, when plants start to get ready for winter, they start basically reabsorbing their chlorophylls. They want to save that energy, and that's when you start to be able to see the yellows that were kind of hiding underneath. Anyway, this would indicate that the light best for photosynthesis is in this blue-violet or in the red spectrum. Okay, so you don't actually need to see me. Okay, So what this is trying to show is really at a chemical level what is taking place in the chlorophyll molecule. So what happens is when light excites that molecule, and here they're showing that packet of energy, right? The photon that was traveling as a wave, but that photon actually interacting with the chlorophyll molecule. When the pigment chlorophyll absorbs light, it goes from this ground state to this excited state. So we actually see an electron, right? It's getting really excited, okay? This is much less stable, right? Lots of potential energy, but much less stable. When that excited electron falls back to its kind of ground state, to its, right, it goes off the diving board, it releases that energy and goes back to where it's more stable. That excess energy is released as heat and light. And so that's what they're actually showing here is if you took a beaker of chlorophyll and you exposed it to light, it would fluoresce, right? Because that energy, and apparently it would be slightly warmer as well, because that energy when it releases is actually causing this glow, this fluorescence. Okay, so this is what it looks like when we're actually inside of a plant cell rather than just in a beaker of chlorophyll. So what we see here is called Photosystem 2. So let's give you a little perspective here, right? So we're talking about the light reactions. We're inside the chloroplast and actually in the thylakoid. So notice, right, the thylakoid was those little discs. We're inside of the membrane here. We have embedded this protein that's going to hold our chlorophyll molecules, our pigment molecules. This outer region is called the light harvesting complex, and you can see all of the chlorophyll molecules here. The reaction center houses a pair of chlorophyll A molecules. Now, these guys are special because the way they're held in here, basically, all of these other chlorophylls, when they get excited, right, by photons of light, they just kind of hand that energy, that excitement off to each other. And everyone is handing that energy to this pair of chlorophyll molecules in the reaction center. These chlorophylls, when they get excited and that electron gets bumped to a higher energy state, right, just like this one, they're actually able to hand that electron off to another molecule, right? And so we're just calling that the primary, there it is, primary electron acceptor at this point, okay? So this whole guy together is called a photosystem, right? The photosystem includes the light harvesting and the reaction center complexes. Okay. So what we're going to see happen here is we are going to break down these light reactions in more detail to see where we get the ATP, where we get the NADPH, and where we get the O2. I think we have all the information we need now to get there. So in these light reactions, we're actually going to see this like linear flow of electrons. Maybe this is easier if I just pull the whole thing up. Okay, so let's look at this. What we're going to see here, this is all taking place in the thylakoid, okay? In the light reactions, we have light coming in to the leaf, right? And it's photons of light hitting chlorophyll here in that light harvesting complex, right? In our photosystem, sending all the energy they have to those two special chlorophyll molecules. And then our electron is getting bumped to the higher energy state, and then we're going to pass it through an electron transport chain. We'll come back to that, right? But notice what happens here is as we pass energy and those electrons get bumped, you need to have electrons to get bumped, right? And you're going to run out of electrons unless you have a way to replace them. And so remember, in photosynthesis, right, in the light reactions, we're actually taking water and we're splitting it, and we see it happen here, right? So the water is being split, the electrons are going to enter the chlorophyll molecules to get excited, and the oxygen is released as a waste product. Okay, so this particular photosystem, to start out, is photosystem 2. We'll see photosystem 1 later, Annoyingly, it's the order of discovery. So the primary electron acceptor here basically is going to be a proton that's involved in this electron transport chain. And so as we're passing our electrons down to photosystem 1 and they're going to lower and lower energy states, we can actually utilize that energy to make ATP. This is substrate level phosphorylation. There's enzymes that are using this energy to make ATP. This is the ATP that we saw coming out of the light reaction. But what we don't have at this point yet is NADPH. So let's see where that comes in. So in photosystem 2, we have slightly different chlorophyll molecules, right? But they're getting their electrons from the electron transport chain, right? In photosystem 2, we get our electrons from splitting water. In photosystem 1, we're going to get those electrons from the electron transport chain. But notice, here we go, more light is exciting electrons. The chlorophyll molecules in this light harvesting complex hand them off to the reaction center chlorophyll. And when the energy is strong enough, those get bumped to a higher energy state. We have a really short, at this point, short electron transport chain. And we hand off the electrons to NAD+, right? Here we go, reducing it to NADPH. You always wanted to know where the ATP and where the NADPH came from in the light reactions, and there it is, right? Now, this is, right, it doesn't just look complex, it is complex, but I thought this was a really fun diagram from your book. So notice how this happens. So we got the big old sledgehammer, this is the energy from our photon of light, and in photosystem 2, right, if we kind of bring this all together, we have an electron that's being raised to a higher energy state, right? It then gets handed off down that electron transport chain, right? And they're showing like a little mill here. That mill is actually making ATP. Actually, I may have lied. That might not be substrate. That's not substrate level phosphorylation. We're actually going to see a little ATP synthase going on here. Anyway, we make ATP. Right, that electron then gets re-excited by a different photon of light. So we see the energy coming in from this big sledgehammer. And when it gets handed off this time, it gets put in a little bucket, right? It gets handed to NADP, which becomes NADPH. And pretty cool. So I thought that was a fun little analogy for this, right? A little clearer what's going on there. So flip back and forth in there if you kind of want to make those comparisons. And here is, right, they are actually showing in that first electron transport chain, right, in here where they're talking about a mill, right, in here where we're passing off those electrons and making ATP, we are in fact, this looks very much like it does in the mitochondria. That electron transport chain is pumping hydrogens between the two membranes in our chloroplast, and plants, this is pretty fun, in their chloroplasts have ATP synthase. It's the same protein. Hydrogens are allowed to cross that membrane, and we are making ATP. Okay. Oh, again, so this is kind of what that is going to look like. So here we see photosystem 2. We're splitting the water to get electrons so that as the light energizes our electrons, they can get handed down the electron transport chain, right? So notice all the hydrogens are getting trapped inside the thylakoid here. So this is a little different, I suppose, the direction here. The hydrogens are trapped inside the thylakoid. The only place they can get out is that ATP synthase. And so here's the ATP that's coming out of the light reactions. If we follow our electrons, right, they end up in photosystem 1 where light can energize them again. They get handed off, these electrons get handed off to NADPH, and sure enough, those are both heading to the Calvin cycle, right? So the end products of the light reactions are going to go to the Calvin cycle. Now the Calvin cycle ends up, it is a cycle, right? So that should remind you something of like the citric acid cycle. It's actually able, because what makes it a cycle is it's able to regenerate its starting material as it goes around and around. Now, the difference, big difference here, there's lots of differences. Big difference, Calvin cycle is anabolic. It is building sugars from smaller molecules, right? From CO2 by using the ATP that came from the light reactions and using the electrons from the NADPH. Okay, and so when they're showing a sugar coming out here, right, that is a more complex molecule than CO2. Technically, the molecule coming out here is one called G3P. We'll look at that. Okay, right, so in the Calvin cycle, big picture, we're taking CO2 and we're making sugar. We can only do that if we can reduce the CO2. Here's our electrons to reduce it, and here's the energy to make it happen. Okay, Calvin cycle. Lots of details here. Let's maybe just go more in this realm of things, right? So this is where I was going. What comes out of the Calvin cycle is a sugar called G3P, right? So we don't just make glucose in the Calvin cycle, but we are making a 3-carbon molecule that then can be built into glucose, triglycerides, right, amino acids, other things. So typically we talk about photosynthesis making sugars, and it can, but technically what comes out of the Calvin cycle is a molecule called G3P. Now, what I think is really important here is to realize that we have 3 steps in the Calvin cycle. So this first step is called carbon fixation. And what we mean here is that we are taking atmospheric CO2, right, gaseous CO2, and putting it into a solid form. Okay, that's all we mean by carbon fixation. The reason they're pointing out 3 CO2 here is you can only get a G3P once you've gone through the cycle 3 times. Because think about it, CO2 only has 1 carbon. G3P, we can see, has 3 carbons. So you actually need 3 CO2s, right? You need 3 cycles in order for this to work. But the first step is always carbon fixation, taking a gas and making it a solid. The second step is referred to as reduction. You should absolutely be expecting to see the hydrogens being ripped off NADPH, there you go, sure enough, and being added onto our molecule. Now you can't see those hydrogens in these images, but trust me, they're there, right? So G3P is much more like a sugar. Okay, the third and final step of the Calvin cycle is really meant to regenerate this molecule called RUBP. So RUBP is going to be what accepts the CO2. So look at this, sorry, I'm on the wrong screen. 1, 2, 3, 4, 5 carbons. Notice when we bring a CO2 in, we make a 1, 2, 3, 4, 5, 6 carbon molecule. So that first step, carbon fixation, is really taking RUBP and adding a carbon dioxide to it to get a 6-carbon molecule that runs through the cycle. And so again, you need to regenerate RUBP in order for this to be a true cycle. So really that's your key takeaways there from the Calvin cycle. So again, big picture, light reactions provide the ATP and NADPH. Calvin cycle is able to utilize those to run the anabolic reaction of taking CO2, reducing it to make sugars. Okay, so we're getting close here, just a few kind of side things about photosynthesis, and we're done. And so, sorry, way too much text on here, but the point here is that we're going to see that dehydration is problematic. So whatever, let's go to the next, I can show you this on a picture. So what they're trying to show you here is kind of, again, leaf anatomy. What we're going to see is there's a few different ways for photosynthesis to take place. So the problem with arid environments, right, is that plants have all those little pores to let CO2 in and oxygen out, right, the stomata. And so what happens is if plants are in a really dry environment, they have to close the stomata to hold on to water, right? But then that's going to actually make it harder for them to do photosynthesis, because what's going to happen is, right, it reduces their access to CO2. CO2 can't come in, and oxygen is going to build up. And this is hard for us to picture, but oxygen is a toxic product, right? So plants are actually trying to get rid of that. If they don't, it sets up this process called photorespiration that's it's not exactly like the Calvin cycle running backwards, but it's basically they start using that oxygen and breaking the sugars, right? So this is wasteful. It's not going to help a plant succeed. And so plants have a couple alternative forms of photosynthesis. So this one is trying to show what's going on in something called the C4 pathway. So normal photosynthesis would be referred to as C3 photosynthesis. And what we're going to see is these two alternative forms of photosynthesis to deal with this problem of dehydration. So C4 and CAM photosynthesis. So the big deal here is when we were talking about C3 respiration, we were just looking in what's called a mesophyll cell. Okay, but in C4 respiration, instead of just using this mesophyll cell, these plants are going to separate photosynthesis into two separate cells so that they can limit the risk of photorespiration. So the big thing to me to think about in here, what we're going to see in C4 respiration is two separate cells. So it's a spatial separation of the steps in photosynthesis. What we'll see here in CAM photosynthesis, they're showing a temporal separation, right? Meaning separated by time, night versus day. So in C4, what we see, right, is CO2 coming into the cell, and it's actually getting converted, right, into a 4-carbon molecule in the mesophyll cell, right? The mesophyll cell then allows that 4-carbon molecule to move down into the bundle sheath cell where the Calvin cycle takes place. But basically, we don't need to have any oxygen down in this bundle sheath cell, and so we can avoid that risk of running photosynthesis backwards, of photorespiration, of taking these sugars and breaking them down, because we've separated this into two separate cells. Again, that's your key takeaway. C4 photosynthesis avoids dehydration by separating photosynthesis into two separate cells. For CAM photosynthesis, it's a very similar kind of idea. Instead of using that malate, that 4-carbon molecule, it produces a different organic molecule called crassulacean acid. So CAM stands for crassulacean acid metabolism. And so basically it's producing that crassulacean acid, and then during the daytime when there's lots of light, it's going to use that to run the Calvin cycle, but it opens up its pores, its stomata at night, and lets the CO2 in. So anyway, plants have some nifty adaptations to survive their environment. They can't run away, and so they have to kind of be prepared. Okay, and so, we're in it. We're in the review. So notice, again, CO2 coming in, oxygen leaving. CO2 comes from the atmosphere. Some people forget this. Water. It's coming all the way up through the root system into the leaves. The product of photosynthesis, the sugar, right, maybe that's eventually made, can then be moved throughout the plant wherever it's needed. If it's making fruit, seeds, it feeds the roots. And this is maybe my favorite part. Those sugars that are going down to the roots actually feed the microbes in the soil and make sure that the plants have the right microbes, microbial associations that they need to thrive. I think that's pretty fun, and people don't often talk about that the plants are actually feeding the soil microorganisms. Anyway, okay. Again, water is being split in the light reactions, which included Photosystem 2, the electron transport chain, Photosystem 1, and the final electron transport chain, right? We're producing ATP and NADPH, which are going to go into the Calvin cycle, into our 3 steps where we do carbon fixation, reduction, and then we regenerate RUBP. The ultimate goal is to reduce CO2 into a sugar. And we're done. We'll see you guys in class.